Hi, I'm Paul Spruell. I'm an associate professor in biology at Eastern Washington University. And many of you, like most people, might be surprised to learn that not only do the students who work with me and I spend a lot of time here in the molecular ecology lab, but we also spend a lot of our time in areas like this doing field work. And for us, we need to be able to do both because much of the work that happens in my lab uses patterns that we detect in DNA molecules to help us understand the processes that are important for the ecology, management, and conservation of native species of fish. Just to give you a quick example, we're currently working with the Kalispel Tribe of Indians, along with the Washington Department of Fish and Wildlife and other collaborators to try to understand how dams like Albany Falls Dam that I have here in this picture, how dams on rivers are affecting native species like the West Slope cutthroat trout in this picture. And to do that, we use exactly the same sort of technology and analyses that a company might use if they were trying to determine your ancestry. And so here I have a map up of my own ancestry. And in order for this company to understand or determine that most of my genetic material is similar to that from Scotland, along with some other countries, as you can see here. In order to make that determination, they needed to be able to differentiate the typical genetic pattern of individuals from Scotland versus the pattern that you might see from somebody from Italy or Spain or Portugal, for example. We do exactly the same thing, except rather than looking at different countries, we look at different streams. And so in this map, each of these streams we know West Slope cutthroat spawn in, and so we really need to ask the question, can we differentiate the fish in each of these streams to allow us to then determine where those fish below each of the dams in the area of interest are actually from. So to get those samples that we need to characterize the different populations, we go out into the stream and collect fish. Here you can see my graduate student Craig Wells, who's using a backpack electrofisher to collect small fish from this stream. And the unit Craig is wearing passes an electric current through the stream, which stuns any fish that he might encounter. Those fish then come to the surface and he will net them and allow us to collect them. After we collect the fish, we then just take a small fin clip. Here Craig's taking a clip off of the caudal fin or the tail of this fish, and it may amaze you to know that this little fin clip that's about the size of your pinky fingernail or a paper hole punch is more than enough tissue to do all the analyses we need. That will grow back, and the fish is released without harm. Well, hopefully that gives you at least a little bit of an idea of the sort of projects that we work on in my lab. I did want to make sure I gave credit to Colin Hendricks, who is an undergraduate who has been working with us all summer. You didn't see Colin in the videos, but Colin was the one actually filming the videos of Craig doing the electrofishing and taking the fin clips. And I just wanted to point out that we have a lot of undergraduates that work with us throughout the year. If you've got any questions, I'd certainly love to hear from you. You can email me at pspruell@ewu.edu, and I look forward to hearing from you.